So this week we'll be doing threats to prenatal development. And as part of this lecture, we'll be looking at chromosomes, DNAs, and genes. We'll talk about genotype and phenotype. We'll talk about the pattern of genetic inheritance and some threats to prenatal, perinatal, and postnatal development. We will equally look at chromosomal and sex-linked disorders and some environmental threats, as well as their educational implications. So let's begin with chromosomes, DNAs, and genes. Every human being is made up of numerous cells. Now, when you take one of these cells and you isolate, it has a nucleus. So from our secondary school integrated science, we know that every plant and animal cell has a nucleus. And within this nucleus is what we call chromosomes, which are thread-like substances that are contained within what the nucleus. Now, the chromosomes contain information that gives the genetic material that every individual or every species is made of. So when we take the human being, human beings have 46 chromosomes, which is made up of what? 23 pairs, one from the mother and one from the father. Now, the first 22 of these chromosomes are called autosomes, and the 23rd chromosome is called the sex chromosomes. Now, when it comes to the sex chromosomes, females have XX and males have XY. I think we talked about that when we were doing conception and prenatal development. Another term we want to look at is a karyotype. What is a karyotype? A karyotype is a picture of chromosomes from a single cell, and it is used to determine possible genetic disorders that a person will have, and then the sex of the individual. So when you take a typical karyotype, you should be having 44 autosomes and 2 sex chromosomes. So a normal karyotype is supposed to have 22 pairs of autosomes, that is 44, and then 2 sex chromosomes. So I have a picture here depicting what a chromosome looks like. And so within this image, you will see that there is a dark chain of what? Molecules within the chromosomes. And that is what we are calling the DNA. So I have a picture here also of a normal male karyotype and a normal female karyotype. So the DNA, or the deoxyribonucleic acid, is a long chain of molecules that contains all the information that a person needs to function, for their cells to function appropriately. Now, the DNA contains specialized instructions that make every person unique. Now, the DNA even of twins might be identical, but they might not be the same because what? There are little differences. And so that is why when we want to do paternity tests, we want to find out who is the father of the child, we want to do what? A DNA test. And so DNA of every individual is unique and different from another individual. Just that in case of identical twins, or in the case of a father and a child, there will be what? Some level of what? Similarity in terms of DNA. Now, when we come to a gene is the fundamental unit of what? Heredity. So we are talking about a specific section of the DNA, and it tells the cell how to make a specific protein, and proteins are what is used by the cell to perform functions to grow and to survive. Now, another term we will have to look at is an allele. An allele is what? one of two or more versions of the same gene. Now, the way genetic inheritance occurs, you usually have inheritance from both the mother and the father. So for every trait, we have one allele from the mother and one from the father. So if two alleles are the same, we say that the individual is homozygous for that particular trait or gene. But if the alleles are different, then we say that what the individual is heterozygous. As we go further, I will explain with diagrams and you will understand. So I have this diagram here of a human being made up of numerous cells. When you take one of the cells and you isolate and you look into the nucleus, you have what we call the chromosomes. Now, within the chromosomes are long chains of what? Protein that we are calling DNA. And a particular section of the DNA is what we are calling what? The genes. Now let's look at the terms genotype and phenotype. When we say genotype, it's actually your unique combination of genes or your genetic makeup. And your genetic makeup is actually not visible. It is something that is internal. And so your genotype is a set of instructions on how your body should use certain proteins and how your body is supposed to function, how it's supposed to be built, etc. Now, phenotype, on the other hand, is the opposite of genotype. And so phenotype refers to the expression of what? The things or the instructions within your genotype. And so I say that it is an individual's observable traits such as height, eye color, blood type, etc. And remember that it is not every instruction in a person's genotype that is expressed or carried out. So I tell my students that it is not scientific to just look at a person's appearance, tell me whether somebody was capable of giving birth to that person. Because the only thing that can prove that is a genetic test. Because we are saying that not everything is in our genetic makeup is actually expressed, it's actually visible on the outside for others to see. So it is only those traits that are dominant that are expressed for us to see. So it is possible for a dark person to give birth to a fair person and vice versa. So I have a picture of the genotype being chromosomes, your DNA and the genes, and then the phenotype being the physical expression of the genetic traits that you have. Now let's look at dominant and recessive genes. So a gene or an allele can be dominant or recessive. If an allele is dominant, it means that it can show its effect or it can be expressed in terms of phenotype, even if the person has only one copy of that allele. However, there are times that a person will inherit what? Two dominant alleles, in which case they are equally expressed. And this is what we are calling what? Co-dominance, where a person has two dominant alleles. They are all equally expressed. And in this case, we are saying what? The person has co-dominance for that allele. There are certain alleles that are recessive. It means that for them to be expressed physically or phenotypically, the person needs two copies of that allele. So we know that when two recessive alleles come together, they become dominant. And so the person will need two copies of the allele for it to be expressed. Whereas for the dominant ones, the individual needs only what? One copy. Now, most of the traits or the characteristics that are passed from parents to offspring are done in a dominant recessive manner. And so the traits such as eye color, blood type, etc., are also passed on genetically. So, for example, when we look at eye color, generally there are two types of eye color: brown eyes and blue eyes. Most of us Africans, Asians, Caucasians, etc., have brown eyes. and the white, some whites have what? Blue eyes. Now the allele for brown eyes is dominant, but that for blue eyes is what? Recessive. So when you look at this diagram, let's assume this side is the father side and this is the mother side. So if both the mother and the father give the allele for brown eyes, then the person will have codominant, because we said the allele for brown eyes is what? A dominant allele. But if, let's say, the father brings allele for brown eyes and the mother brings that for blue eyes, then the child will have brown eyes because there is one dominant allele here. And we said that dominant alleles can express themselves with even one copy. And the same situation is what is happening here. But when we come here, the person is having what? Two recessive alleles. So one from the father, the other for the mother. And so when two recessives come together, they become dominant. And so here, the person will have what we call blue eyes. When it comes to blood group, there are three alleles for blood group. We have A, B, and O. The A allele is dominant, the B allele is dominant, but the O is recessive. Now, if a person has blood group A, the person actually has AO. If somebody has blood group B, the person has BO. And if somebody is actually blood group O, the person is actually OO. But because the A and the B are dominant over the O, we end up writing only an A and B or a capital O for the O blood group. Now, given the assumptions that we said earlier, that dominant alleles are expressed with only one copy, and when there are two dominant alleles, they are equally expressed, and that is what we call codominance. And when two recessive alleles come together, they become dominant. The same analogy applies here. So for example, if the father and the mother are having blood group AB, it means that they can have what? The blood group to be A, B, or AB. They can have it to be A because when the father brings A, the mother also brings A. It is A. If the father brings B and the mother brings B, it is still B. And then when the father brings A and the mother brings B, then that will be what? AB. Now the same applies to if the father is AB and the mother is B. Here, if the father brings A, the mother brings B, it is AB. If the father brings B, the mother brings B, it is a B. Then remember that blood group B is actually BO. So if the father brings A and the mother brings the recessive O, then it is what? It is actually AO, but we have A. And so these are the possible combinations of what? Blood group based on what the father has and what the mother has. If we come here, if the father is O and the mother is A, remember that the child can have O because O is actually OO, two recessive O's, and A is actually AO. So if the father brings the one of the O's and the mother brings the O here, two recessive O's become what? Dominant. So the child has blood group O. But when the mother brings the A and the father brings the O, then the child has A. So you can just sit down based on these assumptions and navigate why we are saying that all these possibilities are the chances of the children having these blood types if the parents have these blood types. You can easily match them. Now, the pattern of genetic inheritance. There are 5 major patterns. Now, there are some inheritances that are located on the autosomes. So if there is an inheritance that's located on the autosome and the allele or the gene for that is dominant, then we say that inheritance is what? Autosomal dominant. If the allele for that trait is located on the autosomes but is recessive, then we say what? That pattern of inheritance is autosomal recessive. If the gene for a trait is located on what? The X chromosome, which is a sex-linked chromosome, but is dominant, then we say that that inheritance is X-linked dominant. If a gene for a trait is recessive and is located on the X chromosome, we say that inheritance is X-linked recessive. If the gene for a trait is located on the Y chromosome and is dominant, then we say it's Y-linked dominant. If it is recessive, we say it's what? Y-linked recessive. So these are the patterns of what? Inheritance. There are sometimes that inheritance is located on what? The mitochondrial DNA within the nucleus, the powerhouse of the cell. And that is what? A mitochondrial inheritance. But what is important is what? The autosomal dominant, the autosomal recessive, X-linked dominant, X-linked recessive, and the Y-linked, which will also be dominant or recessive. I want us to talk about gene mutation. When we say gene mutation, we are talking about a change or an alteration in the genetic material or a genome of a cell of a living organism. Now, this change can be transmitted from a parent towards an offspring. So there are some mutations that lead towards genetic disorders or diseases, and there are hundreds of them. But for the purpose of this course, we will look at a few. Now, when we look at inherited disorders, they can be sex-linked. If we say it is sex-linked, it means that it is located on what? The sex chromosomes. Or the inherited disorders could be autosomal, which means that it is located on what? One of the first 22 autosomes. Now, when you take each one of them, for example, when you take the sex-linked disorders, the sex-linked disorders can actually be genetic disorders. That is a change or an alteration in the gene itself. Or they could be chromosomal disorders, an abnormal structure of the chromosome itself or an abnormal number of the chromosome itself. The same applies to the autosomal disorders. Autosomal disorders could actually be what? Genetic disorders. That is, there's a gene on the autosome that has a defect. Or they could also be chromosomal disorders, which means that it's caused by what? An abnormal structure or an abnormal number of the chromosome itself. So when we take this diagram, we could actually have sex-linked genetic disorders or sex-linked chromosomal disorders. And we could have autosomal genetic disorders and autosomal chromosomal disorders. So we'll look at some examples. So I think I have explained this already. Now, let's look at genetic sex-linked disorders. So the first one is what? Color blindness. And it's a sex-linked disorder that is characterized by inability to differentiate or distinguish between the colors red and green, because there is what? A mutation on one of the genes that is located on what? The X chromosome. And this gene actually allows for what? Color vision, for you to be able to see different colors and distinguish between the colors. Now, the Y chromosome does not have this. And so males will be colorblind if the X chromosome that they have inherited from their mothers has a gene mutation which causes color blindness. So I have a picture, and in this picture, you see the normal color and how people with color blindness see it. Remember that there are different types of color blindness. We have deuteranopia and protanopia. So if this is the normal color, this is how people with color blindness will view it. So this is normal vision, another picture, and people with color blindness. And so as you can see, the green and the red may look like orange to these people. So when you ask them to differentiate the green from the red, they might not be able to do so. The same applies to these apples. I want us to look at another condition called hemophilia. We have hemophilia A or B, and hemophilia is called the bleeder's disease. Hemophilia is also caused by a mutation on one of the genes on the X chromosome. And this gene allows for the ability of what? Blood to clot. Now, hemophilia is characterized by inability of blood to clot and is more prevalent in males and can lead to death from internal bleeding because people end up losing a lot of blood when they are injured. And this genetic inheritance happens because in people with hemophilia, their platelet clocks are weak. Now, when you take the human blood, there are three components. We have the red blood cells, the white blood cells, and the platelets. And it's the platelet what that helps in what clotting of blood. So in people with what hemophilia, their platelet clots are weak, and so the plug is not able to form to cover the opening of what any cut or any wound, and that is why their blood is unable to clot whenever they are injured. The third genetic sex-linked disorder we'll look at is ocular albinism. Now, albinism has to do with a lack of pigmentation, and this lack of pigmentation could affect the eye also. And so albinism is not only limited to hair color or skin color. So ocular albinism is caused by a gene defect, and this gene is the GPR143 gene, which plays a role in the pigmentation, the coloring of the pupil of your eye. So in people with ocular albinism, the pupil is not what's pigmented, and so it occurs also more in female. So I have a picture here to demonstrate how ocular albinism may look like, and this is another one. So you see that usually the pupil is supposed to be dark brown or blue, but these people do not have any pigmentation at all. Now let's move to chromosomal sex-linked disorders. Remember that once we say it is chromosomal sex-linked disorder, it means the disorder is located on the sex chromosome and is as a result of either an abnormal structure of the chromosome itself or an abnormal number of the chromosome. The first one is Fragile X syndrome. Here, the tip of the X chromosome breaks, and so it results in severe mental retardation, speech defects, and interpersonal interaction defects. And it occurs more in males than in females. Why do we say it occurs more in males, or the symptoms are more severe in males than in females? It's because males have only one X chromosome and females have two. And so if there's any defect on the single one X chromosome that the males have, then they are going to have more impairment compared to the females who have two, in which case one which is normal can compensate for the defects of the other. So I have pictures that demonstrate how fragile X syndrome looks like. The other condition we want to look at is Klinefelter syndrome. This occurs in males. So males are supposed to have their sex chromosome to be XY. But in Klinefelter syndrome, these people have their chromosomes to be XXY. So they have more feminine features, high-pitched voice, contours, breast enlargement, little body hair. They are sterile because they don't produce sperms and they have very small testes. And they might have some learning difficulties. So I have pictures that indicate the signs of someone with what? Klinefelter syndrome. Okay. Then we have the super male syndrome. In the super male syndrome, these people have XYY instead of XY. So they have more masculine features. So for example, and this happens to males. Their height is above 6 feet. They have slender body during adolescence. They might have severe facial acne and poor coordination. And it occurs in 1 out of what? 900 live births. There is also female, super female syndrome, or what we call trisomy 47, where females have 3 X chromosomes instead of 2. So these people are taller than the average females. They have long legs and body. Sometimes they are emotionally immature, and they also have what? Learning difficulties. But the issue here is that they are fertile. So people with super male and super female syndrome are fertile, but those with Klinefelter syndrome are not fertile. So here you see that this is somebody with what? Super female syndrome, and the person has 3 X chromosomes. These are pictures depicting people with what? The super female syndrome. Then we have Turner syndrome. In Turner syndrome, instead of a woman or a female to have 2 X chromosomes, that is XX, one of the chromosomes is deleted, so the person has only 1 X chromosome. And so the genetic makeup is what? XO. Now, these people are short in stature. Their necks are webbed. They have small jaws. Their chests are broad or widely spaced. And they do not, their ovaries do not develop what normally. So they don't ovulate. They have some slight mental retardation. Now, if they are diagnosed early, they have what we call hormone replacement therapy or estrogen replacement therapy, which can help them develop both breast and even help them to ovulate, which will make them appear normal. So this is somebody with what? Turner syndrome. So look at the neck, how the neck is attached to the body. Look at the chest, how broad and widely spaced it is. And look at the person's height. This is a grown-up with Turner syndrome. Another picture of someone with what? Turner syndrome. So we'll look at genetic autosomal disorders. Genetic autosomal disorders. So these are disorders that are caused by a defect on the gene, which is located in one of the 22 autosomes. So the first one is what we call Huntington disease. And it is caused by a gene abnormally located on chromosome 4. Now, what happens in Huntington? We have what we call degeneration of neurons. When we say a neuron is degenerating, we're saying that the neurons are dying off. And so it produces dementia. Dementia is characterized by a decline in cognitive abilities, sharp decline in cognitive abilities. And aside dementia, people with Huntington disease also have random jerky movements. Usually, this condition manifests around the age of 35. And usually people die within what? The first 12 years after the onset of the condition. So this is someone with what? A Huntington disease. Then we have phenylketonuria. So it is also caused by a defective gene on chromosome 12. Now, in people with phenylketonuria, or let me say in children with phenylketonuria, because it affects children the more, they are unable to metabolize or break down a type of amino acid we are calling phenylalanine. Now, phenylalanine is an amino acid that the body is supposed to use. But in people with what phenylketonuria, this is not used. The body is not able to break it down, and it accumulates or builds up in the body and causes what brain damage. So PKA can lead to what intellectual disability, seizures, behavioral problems, mental disorders, etc. There is treatment that is available for children so that at least they can have average intelligence. So these are children with what? Phenylketonuria. Then we have resource factor incompatibility. So when you see your blood group, you notice that in writing it, they either attach what a positive or a negative sign to it. The positive or the negative sign actually refers to the what? The RH factor. Now, your blood group can either be RH positive or negative. Now, the presence of an RH factor means that there are certain antigens that are within your blood. So there are certain proteins that are located on the red blood cell of every person's blood. And these are what we are calling the RH factor. It can be positive or negative. Now, RH factor is only significant with respect to pregnancies when an RH child is born to an RH mother, predisposing her to RH disease. What simply we are saying is that sometimes somebody who is, say, a lady who is RH negative, okay, so let's say the person is A negative, marry someone who is actually A positive. Now, what happens? During their first pregnancy, everything is okay. But in subsequent, but during birth, the child's blood is likely to mix with what the lady's blood. And once the woman's blood sees the RH positive, proteins in their system, they'll consider it as what? A foreign material. So they'll produce antigens or antibodies to attack that. And so with the normal pregnancy, it's okay, but with subsequent pregnancies, anytime the baby is formed, the RH positive mother's blood will produce antigens that will attack the baby. But there are treatment options to prevent this from happening so that the child does not lose so much blood and does not die because of this reaction. All right. So this picture basically describes what happens. So a father is RH positive, mother is RH negative, who is carrying a baby that is RH positive. And because during earlier pregnancies, the baby's or earlier deliveries, the baby's blood mixed with that of the mother's. The mother's system is now producing antibodies to attack this baby. And so there are injections that are given to the woman to prevent the woman's body from making these antibodies to attack the baby. So basically, that is what is explained here. The baby is RH positive, the mother is RH negative, and is making what antibodies to attack the baby. Now let's talk about sickle cell. Sickle cell is a genetic condition in which red blood cells are not shaped like they should be. So Normally, the red blood cells are supposed to be in the form of a disc, but they are crescent shaped in sickle cell. Now, sickle cell results as a because there's a mutation on one of the genes that's located on chromosome 11. Okay, and this codes for hemoglobin protein. So in people with sickle cell, their red blood cells are crescent shaped and they get stuck together and they block the small vessels. So ideally, this is how the red blood cell is supposed to look like. But in people with sickle cell, it is a crescent shape, and so they'll block the arteries or the blood vessels. What are the signs of sickle cell? We have anemia. Because the red blood cells are weak, usually they break apart easily and die. And so once you are having fewer red blood cells, it means that your blood count also will be low. Sometimes the people experience periodic episodes of pain because of what the blocking of what the blood vessels. So they have sometimes pain crisis. Now, sometimes they also have swelling of hands and what feet. Now, a child can only have sickle cell if they inherit sickling genes from either parent in an autosomal recessive manner. So what are the genotypes? We have AA. If you are AA, it means you are normal. If you are AS, it means you are a carrier. If you are SS, it means you have what sickle cell. Then we have CC. Now we have sickle cell disease and what hemoglobin C disease, where hemoglobin C disease is also as a result of abnormal structure of what the red blood cell. So in CC, it actually means that the person has hemoglobin C disease. Then AC, the person is a carrier for hemoglobin C disease. Then SC means that the person has both sickle cell and hemoglobin C disease. So you cannot have sickle cell unless you inherit it from your parents. So it happens in what a dominant recessive manner. So when you do the matching, AA and A, remember that the father is giving one allele, the mother is giving one allele. So if both parents are AA, they are good to go. If one is AA, one is AS, they are also good to go. If one is AA and one is SS, they are good to go. If one is AA and one is AC, they are good to go. Even if one is AA and the other is CC, they are also good to go. But ASAS, ASSS, from here downwards, it is not advisable. So one of the things that is advised is that once you're in a relationship, you know the genotype of your partner so that you don't invest so much time. And then when it is time for marriage, before you be doing these things and you find out that you are incompatible. Then let's talk about albinism. Remember when we were talking about ocular albinism, we said that results in a lack of what pigmentation, either little pigmentation or no pigmentation. So here, it is this albinism we are talking about is that of the hair and the skin, and it's caused by mutation in one of what the genes that are located in what one of the autosomes. Now, depending on the gene that is affected, then we'll have the type of what albinism that is in question. Remember that albinism is also inherited in what an autosomal recessive manner. So you can have two parents who are not albinos giving birth to an albino because they are carriers. So if you see this diagram, this woman is unaffected by a carrier. This man too is unaffected but is a carrier. So if this one brings the normal gene and the normal gene, you have a normal child. If one brings the gene for albinism and the other one brings the gene that is normal, they have a child that is a carrier. But in a circumstance where they all bring the gene for albinism, then they have a child with an albino. Let's talk about the types of albinism. So we have what we call oculocutaneous albinism. It is what the very commonest types because the person has inherited what 2 copies of the mutated gene, and it is as a result of what a mutation of several genes which are labeled OCA1 to what OCA7. What they do is that they cause a decreased pigmentation. So there's your skin that don't actually have a pigment or a color, and then your hair and your eyes as well. So you are likely to have what vision problems. Okay. Then we have other albinisms that are related to what syndrome. So a syndrome is a collection of what symptoms or conditions. So, for example, the Hermansky-Pudlak syndrome is a form of OCA, but in addition to the OCA is what oculocutaneous albinism. In addition to the OCA, these people have bleeding and what bruising problems, lung disease, and bowel disease. Then we have the Chidia-Kagashi syndrome, where the person has OCA as well as what other immune problems of recurrent infections, neurological abnormalities, etc. Let's look at the last group, that is chromosomal diseases. So we want to talk about chromosomal autosomal disorders. Here, let's look at Down syndrome. Down syndrome is also called trisomy 21, where the person has what? An extra copy of the 21st chromosome. There are different types. The trisomy 21, the person has an extra chromosome in all the cells. There's an extra copy of the 21st chromosome in all the cells. In mosaicism, the person has what? Extra chromosome in some, but not all of the cells. Then in translocation, they have only what? An extra part, not the full chromosome, an extra part of what? The 21st chromosome in addition to what they have. So people with trisomy 21 will have the severest of the symptoms, followed by mosaicism, and followed by what? Translocation. So I have a picture of a child with Down syndrome with a mother. Similar one here. Now, let's look at all that we have been looking at so far is the environmental threats. Let's look at all that we have been looking at so far is the genetic stress. So let's look at environmental stress. So we know that harmful substances from the environment can also affect the child. It's not only genetic mutations. So these harmful substances are what we are calling teratogens. Teratogens can damage the embryo or the fetus during the critical periods of development. So certain pollutants or toxic waste from the environment can be toxic to the child. Okay. Now, the harm caused by teratogens are not what? Simple, and it depends on the following factors. Those. So, for example, when we take illicit drugs, there are some people who will be smoking for long, nothing will happen to them. But another person will also smoke for the first time when then will have certain effects. So larger doses over a long time periods usually have what? More negative effects. Then we have heredity, our genetic makeup. That is what I said, that there are some people, their makeup, they will take the illicit drugs, they'll be exposed to all the harmful substances, but it will not have any effect. But another person, to be the tiniest exposure, they are affected. So the genetic makeup of the person plays a role. Then we have other negative influences. So, for example, if you are even abusing drugs, and in addition, you are not eating well, you are not going for medical checkup, all these things can worsen the effect of what one teratogen. Then age. The effects of teratogens vary with age of the organism at the time of exposure. So we are talking about critical periods. So there are certain times that teratogens can have more effects than other times. So what are these environmental threats? We are talking about prescription and non-prescription drugs. You just go, maybe you are pregnant, you just go to the pharmacy and go and buy any drug. This is not allowed. Because even when they are advertising for drugs, there's this phrase that they say that it's not good for pregnant women and lactating mothers. And so it's not every prescription drug that you're supposed to take when you're pregnant. We have illegal drugs. So heroin, cocaine, marijuana, we have tobacco, alcohol, radiation, environmental pollution from chemicals, mercury, lead, etc. Then maternal disease, if the mother has HIV and any of the STIs. Other environmental factors are exercise. Some people, when they are pregnant, they don't eat well. A lack of proper nutrition can also affect the health of what the developing child. We have exercise. Some people don't also exercise well. We have drugs, poverty. Poverty because if you don't have the financial means, you might likely not to be eating well, and that can also affect the health of the child. And then the mother's emotional state. And that is why we advise that when women are pregnant, their spouses should not take them towards emotional torture, because whatever emotional challenge the mother faces, it's likely to affect the child. Then we have the mother's age. Certain conditions have been linked to the woman being having the child at an older age, etc. Now, one of the teratogens I wanted us to talk about is alcohol. Alcohol, a lot of people seem to underestimate the effect of alcohol, but exposure to alcohol can result in varied abnormalities. We have what we call fetal alcohol syndrome. Now, there are different types. So we have the fetal alcohol spectrum disorder, the partial fetal alcohol syndrome, and alcohol-related neurodevelopmental disorder. So when you take fetal alcohol syndrome, it's a range of physical, mental, and behavioral outcomes caused by what? Prenatal exposure to alcohol. What happens when a child is exposed to alcohol? The child has slow physical growth. They have facial abnormalities. So they have short eyelids. They have thinner upper lips and smooth or flattened face. And then they have brain injury. And so usually their head becomes very small, and they have problem with memory, language, communication, attention. They have hyperactivity issues, planning, reasoning, motor coordination. In fact, there are a lot. So I have a child with fetal alcohol syndrome. Another picture depicting fetal alcohol syndrome. So when we take partial fetal alcohol syndrome, it's characterized by two of the facial abnormalities, brain injury, and they have at least 3 areas of impairment. So mothers of children with partial fetal alcohol syndrome drank alcohol in actually small quantities, and this exposed the children. Now, sometimes it's not only the mother's use of alcohol. Even if the father uses alcohol as at the time of conception or as at the time of having intercourse, it can influence or alterate the sperm and can have effects on the child. Then we have alcohol-related neurodevelopmental disorder. Now, here, there are 3 areas of mental functioning that are impaired, despite the person having a normal, typical physical growth or the absence of what facial abnormalities. So here, the children do not have these facial signs for you to see, but they have what cognitive impairment. So prenatal alcohol exposure causes this, and this one is less pervasive than the partial fetal alcohol syndrome. And so in your discussions, I would want you to talk about some of the educational implications. If you're having a student in your class with ocular albinism, what does it imply? If you're having a student in your class who has Down syndrome, how do you tailor your teaching to suit this child? So I want you to discuss this in your groups or with your learning partners, and we see what best ways you can apply this knowledge in your teaching. Thank you.